I'm Dr. Ben Newman. I study coronaviruses for a living, and I'm going to try to use that to answer your questions. So, the next question is actually a really good one, and this is from Daniel. This is some big brain stuff. Yeah. So, the question is it possible to make a universal SARS vaccine that targets the core of the virus, which would be the part that doesn't change as much? And then we wouldn't need to develop future vaccines like we do for the flu vaccine and everything else. So if the answer is yes, why hasn't that been done, or when will it be done? Great question. Yeah. So, yeah, all right. Let's unpack. So this is, first of all, like the Holy Grail. This is the silver bullet. This is the grand prize in the science Olympics. Yeah, gold medal times, I don't know, whatever. Yeah. This is what a lot of people would like to be able to do. And there are ways that we might be able to do this, but we do not have a way to do this at the moment. The first thing I'd say is that one thing you can do, well, no. Before we go into the solutions, let's look at the problem. So the problem is that in the human immune system, your immune system sees a virus infection one tiny piece at a time. So what that means is you'll have a region of maybe, let's say, 10 amino acids. This is 10 little building blocks, 10 little Lego bricks out of the entire virus. Virus the size of SARS coronavirus 2 is going to encode about 10,000 Lego bricks, 10,000 amino acids worth of protein. So you can only see this entire 10,000 in terms of little tiny patches, maybe 10 or 20 at a time. And if there's a change to one of those patches, then the antibody or T cell receptor, whatever is recognizing that little piece, may not bind anymore, because it's just going to bind by shape. It's looking for a particular shape that it can grab, and if your shape has changed, then boop, 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 yeah, just may not be able to bind. So that's the main problem. The immune system acts like a person that can't see. It's just feeling around and it's sticking to things. There's no particular intelligence there. It is just a way to make things that stick to other things in such a way that you don't get sick. That's super simplifying it, but yeah, that's kind of all that's going on at at the level of, I don't know, physics. Yeah, molecular biology. The how is much more complicated, but that's the what. Yeah. So, how could we maybe do better? Well, one way to do it would be to pick a part of the virus that doesn't change as much. And instead of putting in the spike, which changes quite a lot because the spike sticks out of the virus, and the parts that stick out are subject to selection. The immune system will kill all the versions that it can, and you may only be left with the ones that the immune system doesn't have a good way to kill. And so by doing that, you've selected this virus, and then this one goes on to start the next infection, and now it's that much harder to kill because all these things don't work. Yeah, so that's a concern. But overall, I'm not. It's been very difficult to find something which the body makes a very strong response to, and which seldom changes in the virus. The closest you get is probably the nucleoprotein. So you can't get any really useful antibodies against the nucleoprotein because the nucleoprotein is never going to be in a place where an antibody can do any good. The only way an antibody binds to the nucleoprotein is if the virus has exploded, at which point the virus isn't going to start an infection anyway. It's a dead one. So yeah, why make those? But you can make what are called a T cell response. So this is a kind of white blood cell, and it can look for parts of the N protein, and it can go in and find those and, yeah, get them. We have really good catalogs now, though, because there are parts of the virus that change a lot less than the N protein, and so why not use those? We have good catalogs from the insides of people, and we can see what it is that they were able to respond to, or what they even should be able to respond to. And the answer is, it ends up being pretty much three things that make up 90-some% of all the response. Spike protein's almost half of the response. N protein, that's like another third, and M protein is in that same vicinity, yeah. M and N are both, I think, 20-ish% of the response. So, put those together, you've got a great response, but M the part that we make antibodies to is a part that can change, so that's not great. Spike definitely changes. That's under the most selection, and it seems to be able to change. The N protein changes some, not as much as the spike, and the changes seem to be in the front half of it rather than the back half of it, for what it's worth. But there again, yeah, I don't know. It doesn't look like universal vaccine kind of territory. So there are people that are trying to make versions of the vaccine that just have maybe sort of, like you could put M and N in there because they change slower than the spike. That's a reasonable way to approach it, but you would probably get less protection due to antibodies, because the antibodies that you make that are going to prevent you from getting infected in the first place pretty much have to be against the spike. And all the parts of spike that don't change are located kind of on the inside where an antibody can't get to them to do any good. So that's annoying, right? But on the positive side, we've kind of already done this. And those are the vaccines that you're looking at now. And they seem to work against a lot of different variants that we have out there. So this is a theory that if you pick an early version of the virus, it's still going to have a lot in common with all of the later versions, because all the later versions came from the early version. They do this with flu vaccines sometimes. Each year they'll look at it, and if they don't have to change, they won't change. So sometimes you'll get like the 09 pandemic California strain of flu being used for years and years. I think that thing's been used for 10 years or something since then, because although the virus is kind of broadened out and you have all these other things, if you vaccinate with the thing at the bottom of the family tree, you still get real good protection against all the little children. So yeah, vaccinate against your elders and you'll protect against the kids. Yeah, that's basically the approach. And so that is the approach that all of the currently approved COVID vaccines have taken. They've used the original version of the coronavirus spike, and they've grown up a bunch of this, and they say, all right, we're going to show this to the immune system in the hope that this is going to be kind of in the middle of the cloud of viruses that, variants that have developed, and all the things that could get you out there. So the other nice thing is that the technology that gives us these vaccines, these mRNAs, we are just vaccinating with biological information. And it is easy as pie to, if there's a different strain and you're not getting much protection with the original version, you just split it. You put in half original version, original recipe, Colonel's recipe, and you put in half new version, and that's now the vaccine. It's got the same amount of mRNA, but you've just broadened what you can make a response to. And that is another totally valid approach, and the way all of the vaccines, even the adenovirus ones, have been made, it's really easy, because the only part you have to change out is you have to put in a different version of the spike gene, or you have to mix half of this one, half of that one. Things like the papillomavirus vaccine, the HPV vaccine, they will frequently mix 4 strains, and it's no big deal. You put them together, you get a little less response against each one individually, but you get much broader protection against a big group. So, yeah, I think a lot of people would like to do exactly what you're proposing and make a vaccine that protects against all versions of SARS coronavirus, past, present, and future. I think that is worth pursuing, and I think with the kind of vaccines we're dealing with now, that is a goal that actually is more realistic than it's ever been before, because we don't need any special new breakthroughs. We just have to keep, finger on the pulse and keep an eye on what the virus is doing out there. And when it starts to change, we just update this thing, and boom, you got yourself a future-proof vaccine concept. So that's how I think it's going to work out. That's how I hope it's going to work out. And time will tell. But generally, I'm pretty hopeful about this. So thanks very much. That was a really good question. And I hope that answer did something for you. I hope that helped and we didn't get too technical. Happy to help anytime I can. And this has been Ask Dr. Ben.